Okay, great. Well, good afternoon for most people on this call, I think. It's a good morning for me. But today it is really my honor to be able to introduce Professor Helmut Grubmüller. Helmut got his diploma in physics in 1990 at the Technical University of Munich, working with Klaus Schulten, who was also my PhD advisor, kind of fun connection. He got his PhD in 1994 and then was a postdoc in the Taven lab until 1998. And after that, he began his position first as the head of the Theoretical Molecular Biophysics group at the Max Planck Institute in Göttingen. He has also risen further in the ranks. He's now the director and head of the entire Theoretical and Computational Biophysics department. And along the way, he has a really long and distinguished track record of using molecular simulation and theoretical approaches to understand the structure and function of biomolecules. Today, I'm really excited to hear about, I think, several stories. We'll see some really beautiful work on uniting timescales in experiment and simulation, on integrating a lot of different data types to look at how ribosomes function and how certain antibiotics work, and some thoughts on the implications of dynamics for protein function prediction. And so with that, I'm very happy to be able to welcome Helmut and really excited to see his talk. Just as a point of order, Helmut has indicated we can take questions throughout, and there will also be a session for more questions at the end, and I will be monitoring the chat. So if anyone puts questions in there, I will raise them at an appropriate time.
Alrighty, wonderful. Many thanks, Peter, and many thanks for a great introduction, which raises the bar of expectations quite a bit. So let's see how far we can get. I should probably first make sure that we really talk about the same thing and on the same page. I'm aware I'm not the only one reporting on molecular dynamics simulations, but I should like to give a very brief intro and then we move into several topics, as Peter already mentioned. So first of all, I also want to acknowledge all the people who contributed to what I'll be reporting on, here shown on the slide. And I will be mentioning names along, and a few are also on the market. So if you feel that you need a great faculty member, just let us know. Right, so to start with, I just want to see that we agree on the fact that proteins are not just simple molecules which have some properties or observables you can measure. They are in many cases literally molecular machines, by which I mean they have moving parts, and it is the motion of those parts, which we call conformational changes or conformational transitions, that actually facilitate the function of many of those machines. And so I think you are motivated that it would be a great idea to really look as closely as we can into the motion of these parts. And that is where molecular dynamic simulations, of course, have their place. And just to be sure, by motion, I do not mean the thermal wiggling of all the atoms, which occurs on pretty fast picosecond timescales or even faster. I do mean the typically much slower, really larger concerted structural transitions, which can occur on nano, micro, milliseconds, you name it, up to protein folding, of course. So that's what we are interested in. And to be very specific, although I will not talk about this wonderful machine today, but I think it's just a wonderful paradigm of what we mean when we talk about molecular machine. And we have worked a lot in my lab also. And also Klaus Schulten has worked a lot on the F-ATPase. And I'll specifically focusing, I hope you can see my cursor on the upper part here. That's called the F1 part. And if you look at it from the top, you can see it's actually a hexamer, and it contains literally a axle, which is very much like the crankshaft in an auto engine, like this one, right? So it rotates actually, and it is this rotation which transfers energy from the lower part here, that's the FO part, which in turn is, by the way, driven by a proton flux right through this red part here, between the red and yellow part here. So this is driven in rotation by an osmoelectric force, and delivers the energy up there where the E ducts ADP, adenosine diphosphate, and phosphate can bind, like shown here in this binding pocket here. And after a rotation of 120 degrees of this orange gamma subunits, as it is called, actually a ready product molecule, the ATP, is released or set free. This is actually the energy consuming step. And so you have a mechanical, chemical energy transduction. And to me, this is one of the really prime examples of a molecular machine. And again, it is really like a car engine, like the Otto engine in your car. It has a rotating crankshaft. It has pistons moving back and forth in the cylinder. That hasn't probably become clear, but these parts here, they move out and in, driven by the eccentricity of the gamma subunit. So they very much resemble the cylinders. The only difference being to a car engine that this thing can run forward and reverse, by which I mean it can be driven by ATP and then you drive this rotation, like that's how a car works, but it can also driven in reverse. You drive the axis and you synthesize ATP. That's the primary function in your mitochondria. And this is not exactly how my car works. I try to push it, but I never get fuel out of the exhaust. So that's not how my car actually works. The reason why this machine can do that and my car can't is actually that my car works at 20%, 25% efficiency, and this machine works at nearly 100% efficiency, which makes us, of course, curious. How can this thing do it? And that's one of the reasons where we look into it. Note the length scale here. We are really at atomic scales, and therefore all our simulations we are carrying out actually are atomistic simulations following the motion of each single atom under the influence of the forces exerted by all the other atoms onto each single atom. So that's what we're gonna talk about. I have a second example of a molecular machine where I actually will be talking about today, and that's the ribosome. It's a much huger machine. In the simulation, it has like 2.2 million atoms, so that's quite large if you want to do many microseconds. And you can also see that, I'll go into detail a little later, but you can already see here how mobile, how flexible this thing actually is. Just look at this so-called L1 stalk here, which seems to kind of grab this orange tRNA, which is bound to the binding pocket here, right in the middle of the ribosome, and then moves over to a different binding pocket, which is called the exit pocket, from which it is released. And if you look at it, that's something I will not go into more detail, so let me mention that briefly now. It seems that the L1 stalk really grabs this orange tRNA and pulls it along, but this is something which structural studies can never reveal. Is it actually that the L1 pulls the L1, the tRNA along, or is it the other way around, that the tRNA is the driving force and pushes the L1 out of the way? What is the driving force? What's the cause? What's the action? That is something our simulations can reveal and experiments rarely. So that is an important point where I think molecular dynamic simulations can play a crucial role to reveal mechanisms and cause and effect chains through the whole molecule. All right, so for those who have not seen, few I think, but nevertheless, who have not seen molecular dynamic simulations, in a nutshell, what we are doing, we tell our computer the position of each single atom in the simulation system. For that one I show here, that is a water channel in a membrane, about 100,000 atoms, including the water molecules. That's this reddish, whitish sprinkles here. And here you can see a top view. And so up to our record is, I think, 10 million atoms or so for a virus capsid, and a record growing. So that we put into the memory of a computer, and then we ask our machine to calculate for us. And I could, of course, spend a whole semester on explaining how. I won't. Here, all the forces which act between all the atoms in this simulation system. Just to mention a few, these are chemical binding forces. Yeah, chemical bonds can be stretched, but of course, if you stretch them, they exert a counterforce. Chemical angles between chemical bonds can be bent away from its equilibrium angle, and then again you get vibrations and restoring forces. Very importantly, Coulomb forces, electrostatic forces between essentially all the atoms, each of which carries a partial charge typically, are very important. Actually, those cause of much of our trouble in the simulation in terms of computer time. So these are the computationally most intensive forces. And there is, of course, Pauli repulsion, which prevents atoms from just moving through each other. That's why we are not just falling through our chairs to the center of the Earth. Luckily, there is this repulsion we also implement. Essentially, we implement everything we know about molecular physics into these potential function, which we call force fields. And once we have these forces, of course, it's a piece of cake to integrate the equations of motions in classical approximations to get actually the motion of all the atoms. And let me make clear one thing. I understand there is quite a bit of students and postdocs in there. There is a very frequent conceptual misunderstanding. We all talk a little loosely about classical simulations, and that seems to imply that we don't have quantum mechanics. After all, there are QMMM simulations, quantum mechanical simulations, and those seem to, of course, capture quantum mechanics. But let me clear about two misunderstandings. First of all, in both, in our so-called force field-based simulations and in the quantum mechanical simulations, the nuclei move according to the classical equations of motions, Newton's equations of motion. So there is no difference between the two. Where the difference is of course, in the treatment of the electrons. And there, the quantum mechanical calculations allow rupture and formation of bonds and allow to include the quantum mechanics of the electrons a little more accurately, but this by no means implies that the force field doesn't capture quantum mechanics effects. After all, a chemical bond is a quantum mechanical effect, and the force field describes a chemical bond quite nicely, except it can't break. But other than that, it fully captures the quantum mechanics of the chemical bond. And so that's just a misconception which I encounter quite frequently, and I just wanted to make use of the time to make that point clear. For those more inclined towards physics, I just want to show here that actually all our simulations at the end are based on the time-dependent Schrödinger equation. It's just we are not able to solve that equations on our current machines. It's huge computational effort, far from being able to do that today, very likely also in 10 years. And so we need to do three approximations, which are Born-Oppenheimer approximation, and then, as I mentioned already, the motion of the atom nuclei are in classical approximation, and then we have this force field approximation, which is essentially just a fit function to what actually we would like to calculate, and that is the ground state energy here as a function of the positions of all the atoms. That is the one which in QMMM calculations is described by explicit calculations and is approximated by a force field. Right, so just to make clear, all is rests on fundamental physics, but involves a number of approximations and assumptions. And with any approximation, sometimes it works, sometimes it doesn't. And you need to develop a kind of a guts feeling where we have to pay attention, where we have to be careful, and which observables can be calculated pretty reliably. But of course, we don't want to only rely on guts feeling. We want also to check. And I always try to teach that we should, whenever we encounter a new system or a new set of observables we want to calculate, first thing to do, compare to experiment and see if it works out. And if that works out, then it's a good idea to move on and to look into things which can't be measured in the simulation. And so that is, these two steps, I think, are always part of, or should always be part of any simulation project. Check against experiment and then look further. Okay, so the raw data we get from such a simulation is what we call a trajectory. This is just, as a function of time, the position and motion of every single atom in the simulation system, which creates these nice movies. I hope the Zoom gets it across somehow reliably. You can nicely see the water molecules actually here going through the channels. Water is red here, blue is the molecule, the protein, and green and yellow is the membrane in which the protein is embedded. And you can actually see water permeating nicely through this water channel here. That is work, very early work by Bertie Groot, at that time postdoc in my lab. And so that would be one example where you can also nicely check, and actually we did that. We just counted the number of water molecules passing through the channel per nanosecond or per 100 nanoseconds, and that gives us a permeability, a permeation coefficient, and that can be directly compared to experiment, and it turns out to be correct within about 20%. Amazing accuracy for a rate. Yeah? That's just a fraction of a KT in terms of barrier height. So that's pretty accurate. And not only is that true for the wild type, we also suggested a number of mutants, and also the changes in permeability due to these mutations actually comes out quite nicely from the simulation. Because the whole thing is mainly governed by the formation and rupture of hydrogen bonds and electrostatics, turns out that these interactions are described pretty well by these simulations. So that is just to get us all on board and on the same page. And I'd like now to start actually with 4 stories, roughly 5, see how time works out. I want first to tell you a very recent example on, I think, a most direct comparison to experiment, in this case, atomic force microscopy experiments. Then I would like to spend a little bit more time to talk about one of my favorite machines these days, that's the ribosome. And then we'll turn our attention to intrinsically disordered proteins, and finally, maybe a more distant view on, if you want, the dynamics not only of one protein, but of the whole universe, if you want, of proteins. Can we maybe have a more distant view of proteins? We call that the dinosaur. So let's jump into it. Ligand unbinding revisited. Very early on, in the mid-90s, Hermann Gaup, who actually pioneered single molecule atomic force microscopy, rupturing single molecules or dragging ligands, as shown here on the right side, these yellow ligands, bioteins in this case, out of the binding pocket of receptors, proteins, streptavidin in this case here in red. That's a non-covalent bond, but it's one of the strongest non-covalent bond known in biology. So these streptavidins bind biotin pretty strongly and very specifically, and so they chose it for as a test system here. And these days they have widespread use in biotechnology. Whenever you want to fix something on a surface or so, you use biotin streptavidin. So how this works is you connect these ligands via a linker to the cantilever of an IFM, which is just a small spring, a Hookean spring, which can be bent, and so you can measure a force with it. You approach it to the surface, and here you can see you retract it then, and you measure a force. So the whole thing, like shown on the image here, is put under tension in between the cantilever and the surface, and eventually one after the other molecule ruptures, and at that point here, finally, just one molecule survives, and the rupture, the drop of the force to 0, indicates the rupture of only one single molecule, and so you get the rupture force. Now, the term atomic force microscope is a little bit misleading in this case. Here you don't see anything, you just measure a force, and so Hermann Gaup and his colleagues were quite curious and approached us whether we might, in simulations, see what's going on when we drag out a ligand from a binding pocket, and that's what we did at that time. And actually, we revealed and discovered that it's far from just being a bound state, the barrier in between, and an unbound state, and that's all. It's much more complex. So as you can see here, this ligand undergoes quite a number of structural changes, and also its position changes stepwise. In the binding pocket, it's stepwise moving out the binding pocket with transient interactions, for example, here to this loop. And also the protein, the whole binding pocket undergoes induced fit motions, or induced unfit motions, if you want, in that case. And all that complexity is reflected in what we call a force profile here. So each of those maxima reflects the rupture of a, for example, hydrogen bond, or a salt bridge, or a hydrophobic patch, or a water bridge. So you have quite a number of interactions in concert acting, and the whole thing unbinds through a whole network of intermediate states connected by rates which are different from state to state, and which also depend on how fast you pull on the loading rate. Right, but of course the question at the time was, how do we compare with experiment? Okay, so we did the comparison, And on the left side, you see the forces measured by AFM as a function of how far they pull, how fast they pull. That's the loading rate. And on the right side, you see what we were able to calculate from the simulation. Mind you, simulations are quite a computational effort, and this single dot here meant half a year of computer time at the time, 20 years ago. So we just couldn't pull slower because we just didn't have the patience to wait for 20 years. And so that is the best we could do. And on the other side here, this dot here from the experimental side, they couldn't pull faster because there's the resonance frequency of the cantilever, and that interferes with the pulling. So that destroys the accuracy of the force measurement. And so you can see, disappointingly, there's a 5 or 6 order of magnitude gap in between theory and experiment, which makes it quite hard to see whether or not the experiment now really agrees with simulation. So I would say it's really a large call. So amazingly, we still made it into science, but at that time, that was really great, but there was some room for improvement. And now, since AFM cantilevers have become smaller and smaller, resonance frequency higher and higher, the computers and our software, GROMACS, has become faster and faster. Now, we thought last year, let's revisit that. And we teamed up with Simon Scheuring, who is one of the masters of high-speed AFM, and calculated a few more rupture forces for the same system, now pulling much slower, 3 orders of magnitude slower. And that's the result. Same curve, but now with our most recent data. And again, blue here, the experiment. And you see now these days we are able to wonderfully overlap with the simulations. Now our simulations even got error bars. We didn't have that before because it's single simulations. Now each point is here 20 simulations, so we can also get a nice variance and an error bar. And that's how it looks like here. And so if we now, I guess you will agree that here now the overlap is pretty nice. And mind you, that is a force field, just off-the-shelf force field, which has never seen an AFM experimental data as input. So it has been never calibrated against any AFM experiment. And yet the agreement without any further fitting parameters is pretty nice. I think that really also underscores the power of these simulations. And secondly, you see now that if we combine experimental data and simulation, we actually get a dynamic range of 11 orders of magnitude, which is quite a bit. By the way, I see 2 hands up here. So maybe I should just stop, and if you want to speak up and ask a question,
yeah.
Go ahead.
Is Prabal here?
Yeah.
Yeah, so that's very nice. So how do you convert this into a free energy landscape? What model is used to do this, or just?
Okay. So I was about to get to that. So far, I haven't mentioned free energy landscapes. All we do is an atomistic simulation, and we calculate the force from it. I'll get to the landscape in a minute. Give me a minute.
And related to this, the simulation, was it in a, so this is a constant force or constant velocity?
Constant velocity, that was.
Constant. And the experiment was also on constant velocity?
Yes, exactly. In fact, at these timescales, the experiments are always constant velocity, even if they say they are constant force, because you have a feedback mechanism, and that can only be as fast as the inertia of the cantilever. So if you go to faster timescales, it kind of blends into constant velocity, even if you think it's constant force. Because of that feedback. Maybe one more thing, the loading rate actually we read out of the simulation, and that is the slope of the increase of the force. There's a simulation data here with time. That's the loading rate, which we plot here on the x-axis. And that's the simulation, the experiment, by the way, and they get the loading rate exactly the same way. So we really tried to mimic the experiment as closely as we could. There was another question by Amir.
Yes. Hi. Actually, like in simulation, we can pull a molecule using this SMD. But I wanted to know, like, how do one ensure in these microscopy experiments, like what kind of bonding is between the tip and the molecule? How do they ensure, like, they are pulling a single molecule or multiple molecules? So,
oh yes, yeah, I was not clear enough on that one. Yeah. So first of all, it's all covalent here to the tip. So the weakest bond is actually the streptavidin-biotin non-covalent bond. So that is okay. How do we know, or how do they know it's a single molecule? That's a very good question. What they actually do, and this is actually historic data, they repeat the experiment thousands of times, and then they do a histogram of the forces. And the peaks of this histogram, that would be single, double, triple molecule rupture. And in an ideal world, the polymers are pretty long and there is no jungle around formed by other molecules. And then if your force jumps from a finite value actually to 0, it's very likely it's one molecule. And if you do the statistics, you can extract that.
And the bonding is covalent between the tip and the molecule, in the AFM tip.
Correct.
Okay.
Correct.
That's I have a question.
I'm not so sure of a chemist, but very often it's a his tag or something like that. And that's pretty strong. There was one more question.
I have a question. This is Debashish. When you have these double ones, so there's a possibility that the one bond which is broken can reform before the other one is broken, right?
That can be, yeah.
So what effect does it have? Because it will also interfere with the rate of ramping, right?
If you have two molecules kind of competing with each other, that's what you mean.
Yeah.
Yeah. But then you would probably not drop to 0, right? You would rupture one and the other would survive, except that the two linkers are exactly the same length by accident. I think these things all get eliminated if you look at the full histogram, right? If you do it in a single case, it may be hard to tell whether it's one or two or whether they interfere. But if you do the histogram, I think that clarifies things. And I'm a bit
my question is that when you are ramping up, if the ramping speed is not high enough, so every time you do it, so there is enough time for the broken one to reform. On the other hand, if you did it very slowly, so that the story would be different, right?
I think I see what you mean, but notice that there is long polymers here. So once this is ruptured, it moves away pretty quickly and the chance of reformation is super small.
Oh, I see. Okay.
Does that address the
question? Okay.
Prabal, did you have anything else? Your hand is still up.
I don't have. I just lower my hand. I forgot. Yeah.
All right. Great.
Thank you.
I think there is someone in the chat box also.
Yeah, I saw that. Is the force applied on a single atom or a bunch of terminal atoms? Oh yeah, in our simulations, the force is exactly applied to this oxygen here. And this is exactly the oxygen which we have also in the experiment covalently collected to the polymer, which goes to the cantilever. So also in that respect, we are very similar to the experiment. Okay. Right. Okay, so what can we learn from these simulations? Well, first of all, as I mentioned, we have this huge range of loading rates for which we have now both experimental and simulation data. And the advantage of that is we can apply a couple of nice theories. We also developed one recently, but others also, Chabo and Hummer, which convert these force curves into free energy curves. Actually, we did two things. We applied this theory, and also we did numeric simulations of the Smoluchowski equation, simple one-dimensional diffusion in this one-dimensional potential. And then we changed the potential systematically such as to get out of this simulation a best fit to this data here. And it's a bit thin, but I hope you can see the dashed line here. That's actually the outcome of these simulations. So what that tells us is that a pretty simple energy landscape with only one intermediate barrier like here can actually describe this whole rupture process. Does not mean that in reality there is only one well, there's many, but we can describe with a simplified model this whole rupture process over 11 orders of magnitude. And I think that's quite remarkable, and it tells us a lot about the depth of the well, about this this second barrier, which actually I'll go back to in a minute, and so on. So this is one thing which we can learn together with the parameters describing this energy landscape, which probably addresses one of the questions we had before. And the other thing we can learn from these simulations is how do these energetics and also the kinetics depend on how fast we pull. It was before already mentioned that maybe we have rebinding when we pull pretty slowly. While this is not so much the case, what we do have is that when we are not completely unbound, then the path through our different minima leading to the unbound state may be different depending on when we pull fast. And this is shown in this simplified graph here. So each metastable state, each intermediate is here shown as one of these circles here. And the lines connecting are transitions, which we see in the simulations. And if the line is thicker, it means we see more transitions. If it's thinner, it means we see less transitions. And if it is blue, it means slow, and red means hot, fast, right? And so what you can see from that plot is, and these are simple representation of how these intermediate states actually look like in structural terms. And what you can see is if we pull pretty fast, then we kind of skip these two intermediates, or this one particularly. So we move from here to here to here, and then very often already to the unbound state, or via this intermediate and then to the unbound state, but only very rarely through this intermediate number 4. That is sparsely populated when we pull fast. On the other hand, if we pull very slow, then actually we go most of the time through all the intermediates, and only rarely we see unbinding. So here we have a heterogeneity of unbinding ways, pathways, which actually depend on how fast you pull, which is also quite nice in terms of connecting to the experimental data. And finally, I mean, agreement with already existent experimental data is nice, but even better is if you can make predictions. And one prediction which came out is here shown on the left side. When we looked at our force traces, sometimes we saw these intermediates at the rupture point very late during unbinding, right? The last step which happened before unbinding was actually this intermediate state here, which you can see here. And that prompted us to suggest to our experimental colleagues, to Simon Scheuring and colleagues, please have another look into your raw data. Maybe you can also find these intermediates. And lo and behold, they did here in red. You can probably hardly see them, but if we enlarge them, zoom in, then you can see these intermediates also in the experimental data. And not only that, also the dwell times in these intermediates shown here on the right plot agree pretty well if you superimpose here now again the experiment in blue and the black triangles are the simulation. And again, there's a nice theory by Dutger and Hammer, and it also fits both the experimental and the simulation data. So again, that is a connection which was not put into, was just a prediction, and turns out to be also pretty accurate. So that gives us confidence that actually that our simulations, at least as far as atomic force microscopy experiments are concerned, are pretty accurate. Right, so that's what we learned from these quite recent simulations. Let me switch gears to ribosomes. And the first story I want to share with you is takes place in the ribosomal exit tunnel. I should probably say, I should have a larger picture, but hopefully you can see it also here on this symbol here. So in the ribosome is a huge machine, and right in the middle of the ribosome, in the core of the ribosome, there's the active site, which is called the PTC, the peptide transfer center, where the amino acid, which is delivered by the tRNA and which is filtered, if you want, by cognate fitting of the base pair of the tRNA on the messenger RNA here on this side, such that the tRNA always carries along with it the right amino acid following the genetic code. So this amino acid is delivered right here at the center of the PTC, and by nucleophilic attached to the growing nascent peptide chain, which forms the translation product, the protein at the end of the day. Before it can form that by folding, it has to traverse a lengthy tunnel, quite a long tunnel here, which goes all the way 30 angstrom through the ribosome, and many antibiotics actually work at this tunnel. So naturally, we were quite interested how actually the nascent chain penetrates through the tunnel. And that is one thing we focused at recently. So this is work by Michael Koller. So we are just writing that up. And in this zoom, you see the active site here on the left side. So this is the PTC. Part of the RNA is the transfer RNA is still visible here. This is the tip of the growing chain. We added a fluorescent dye there because Marina Rodnina at our institute, they are making experiments on that. So we want to also connect to those experiments. And the tunnel here is going to the right side here, so towards this exit sign here. And so what we do in these simulations, we add one after the other amino acid, like in reality, at the PTC, and we see how the amino acid, the growing amino acid chain here traverses the tunnel. And we are interested in that also because somehow biologists don't wonder about it. But as a physicist, I'm really staggered by the fact that what nature is doing here is essentially pushing a cooked spaghetti through a winding maze, right? So a winding road or tunnel. And if you have ever tried that, that's pretty hard, right? So it's super flexible. The persistence length of the peptide is actually pretty short, like 1 or 2 amino acids. And so it's quite amazing that this actually works, and nobody knows how. And so that is one thing we are interested in. So that is ongoing work. What we have already done is and that came out from a collaboration with Dan Wilson and others. He's now in Hamburg in Germany. They made the first cryo-EM structure of a stalled peptide synthesis ribosome and a stalled chain. So in these structures, they were managed to stall it such efficiently that the nascent chain is here visible in green here, right, towards this red thing here. So here the tunnel goes down, right? So you have to rotate your heads now by 90 degrees to the right. So this is now the tunnel, the exit tunnel in this direction downwards. And actually this red small molecule, that is antibiotics, erythromycin. And it is known that this antibiotic stalls ribosomal transactions in bacteria. So that's how this thing works. Quite nice. Luckily, it doesn't stall our ribosomes. That would be bad. And the general belief more or less was, yeah, this is just an obstacle in the tunnel. Sure that this blocks the expression of the peptide through the tunnel, like a cork in a bottle. Simple as is. Now,
Helmut, I think Prabal has a question. If you.
Oh yes, please speak up.
Can I go back to the previous slide?
Yeah, sure.
So here exactly, I mean, so is that this growing chain, I mean, is that done, how exactly it is happening, what technicalities?
Yeah.
For adding monomer after monomer?
Yeah, it's one monomer after monomer. It is not that we mimic or that we simulate the actual synthesis step now with QMMM or something. We just insert more or less manually the monomers. Now we have an automatic procedure for that, but that's unpublished yet, so I can't speak too much about that. But it's really inserting one monomer after the other.
And how do you, I mean, so there is some kind of configurational bias happening? I mean, how do you make sure that no overlap, or how do you take care of the energetics when you insert
we let the additional amino acid simply grow by TI, thermodynamic integration, and that pushes away the rest of the molecule quite brutally. So the idea is not to model as accurately as possible the actual synthesis step, which would be quite challenging, but just to see what happens with the growing chain when it is pushed from behind all the way. So that's what these simulations are.
But how do you take care of the dihedral during this growing, as you adding monomer, the dihedral of the growing end and the dihedral at the other end?
We have a force forcing it into the right dihedral configuration, which we would observe in nature also. And that force, once it's done, is released because sometimes it can flip back and forth. That can happen.
I see. Yeah. Okay. Yeah. Maybe I'll talk to you later. Okay.
Okay.
I'm also surprised. Can I ask this question? Because if this is Debashish, sorry to interrupt.
Yeah.
If this has to move forward in a directional manner, so there is no EF2, EFG, anything of that sort. There is no tRNA movement. So from where is the energy for directional movement coming?
You're absolutely right. There's no EFG, nothing, because again, we don't want to. We have simulations modeling the EFG action, and so that is also ongoing work, but not part of that project. Here, really, we just want to know how the nascent chain traverses the tunnel. And in our simulations, the energy is just put in by the appearance of the new amino acid, right? So that costs energy, and that's how the energy is provided for the translation of the peptide.
So the binding energy released by the synthesis is the driving force?
It's not the binding energy. We artificially appear amino acids, and that's the driving force. It is this appearance which pushes the rest of the chain towards the outside.
Okay.
As I said, quite brutally.
Okay. I'll ask later. Okay. Continue.
Yeah. So I did not super well cover the question, probably.
Well, my question was, why doesn't it go forward and backward? If it is directionally pushing forward, there has to be continuous energy supply. And from where is that coming? So it's not very clear to me.
Can I just ask a clarifying question for Debashish? Are you talking about in the simulations or in the
in the simulation? In the simulation.
Talk about the simulations, yes. In reality, the energy is, of course, supplied by the delivery of the amino acid, and in particular, there is GTP hydrolysis involved. Right. So in EFG, there is the GTP hydrolyzed, which, by the way, is another project, how that affects translation and translocation. So that's where the energy comes from in reality.
Right, and but I'm asking about the simulation. Yeah.
In the simulation, it's the artificial input of amino acids which provides the energy. They have to grow and they have to push against the other, so they have to expand the volume. And essentially, it's if you want PDV, right? So pressure times the change of the volume.
So it's some kind of entropic origin that it is.
That would be enthalpic.
Enthalpic. Okay, fine. Okay.
That would be enthalpic. And it's not only one amino acid, right? We let appear one, then we wait for a microsecond, and then we appear the second one, and we wait another microsecond, and so on. And it cannot go back, right? Because sterically, there is new amino acids appearing, so it can't go back. What it can do, and what it does do actually, is it's curling up. So so it kind of blocks itself. And that is something which does not happen in nature and in vivo conditions. It does happen, though, when we have stalling. And so what we see here is one possible stalling mechanism.
Okay, please continue. Okay, thank you.
By the way, the reason why it blocks in the simulation and not so much in nature we have about 10 amino acids appended per second. Obviously, we can't do that in the simulation. So we have something like 1 per microsecond. And so we are, again, we have to be much faster than in nature, and that's most likely the reason. It doesn't have enough time to equilibrate. Mm-hmm. Right, so back to our antibiotics, Erythromycin. So here most of us and most of the experts believed it's like a cork in a bottle. But there's two findings which speak against such a simple picture. One is that in this density, in the green density, you see a little bit of density already passing by the red molecule. Yeah, you see it goes down to about here. It almost has already passed by, and the question is, why doesn't it just continue? So that is not compatible with a simple blockage, right? And the second, maybe even more important evidence against such a simple picture is that the stalling is sequence specific. So some sequences stall, others don't. And those which stall, you can do mutations and then you unstall this thing again. So you recover translation. And also that if some sequences can go through, cannot be a simple blockage, right? So how does this sequence specificity come about? So these were the questions we were addressing with those simulations. And there was only a structure available with the erythromycin in place, which is the one which you have seen here. There was no structure in the absence of erythromycin, simply because then it doesn't stall and you don't get a structure of the nascent peptide. So the first thing we asked is, what happens if we remove the erythromycin? And what happened was exactly what we expected. So this, in this schematics plot, this amino acid chain, which is growing here towards the outside, moves towards the region which otherwise is occupied by the erythromycin. And so that suggests that erythromycin forces this chain here kind of towards the tunnel wall, and then it undergoes some, or it forms some interactions with the tunnel wall, and that would essentially stall. And that would also explain the sequence specificity. And indeed, so we did have a number of simulations which look like that. In light green and red, that would be, in green, that would be in the absence of erythromycin, in red in the presence. And as soon as you remove the erythromycin, you can see this chain here occupying the volume of the erythromycin, which is shown here on the y-axis. So that works nicely. So how does that explain the escape mutations, the recovery mutations? So some of them are here. So here, number 10, 9, and 7, these arginine in particular here, number 7, that is a strong escape mutation. If you replace it by alanine, the thing starts translating, recovers from the stall. And so those indeed in our hands form very strong bonds, and when we do the mutations, these bonds are relieved. So that is all fine and kind of straightforward. We can understand that. But then there's this single lysine here, number 11, and that really gave us a headache. So why the heck does this lysine, if you replace that with an alanine, also recover translation? That was totally unclear. And Lars Bock was the one in my group who discovered that as soon as you remove the erythromycin, not only does the conformation of this peptide here change, it is also that the conformation of the phosphate backbone of the tRNA here on this side here changes quite a bit. If I zoom into this region, then you can see here the phosphate backbone in red in the presence of erythromycin, and in green in the absence of erythromycin. So as soon as you put the erythromycin in there, the backbone is kind of pushed to the left, and that means away from the PTC. Now, this here with a plus sign, that's the lysine here, and that is an amino acid which is just about to get transferred by nucleophilic attack to the growing chain, which is here connected to the P-type RNA. Here it's the A tRNA, here it's the P tRNA, and the chemical reaction is in this configuration just about to happen. Now, if you retract these phosphates, which are negatively charged, it's plausible that maybe the lysine, which is positively charged, is also retracted towards the left, as indicated here. Compare the red lysine here with the green lysine in the absence of erythromycin. And the distance here to the D10 here is increased significantly. And so we propose that this might be the reason why actually translation stalls here. And actually now, if we look at those distances, for example, here, that would be, well, this would be the backbone position here. You can see a shift, quite dramatic shift here. That would be now the distance between the backbone and the ASP here, ASP10, so that increases. But most importantly, here is this distance between the ASP10 here and the lysine. And you can see this is also increased by, I think it's about half a nanometer, as you can see by these two histograms. And so this increase is real. And if you now change in the simulation the lysine with an alanine, then you can see this effect recovers. So then the two distributions, even in the presence of erythromycin, are nearly indistinguishable. So that explains actually why alanine can recover translation. Now, strangely enough, at that time, nobody had done after this hypothesis, quite obvious test, namely to replace the lysine by an arginine, which also carries a positive charge. And where we also see then such an effect here. So we proposed, let's do this experiment. And our colleagues fortunately did that in Martin's Reed. And there you see that normalized to the stalling effect of lysine here on the left side, the arginine has an even stronger stalling effect, whereas the alanine recovers stalling, no stalling, or very little stalling. And so, yeah, that is again a prediction which we made, which turned out to be correct. And actually, I think provides nice evidence that this mechanism is actually at work. Now, I should like to point out that this is a mechanism, if we now put things together, which involves quite an amazing allostery. So the distance between the binding site of erythromycin and the change of the backbone in the ribosomal RNA is really something like 15 angstrom. And so we have an allostery going on over this distance, and a totally unexpected result, namely that we do not block more or less the nascent chain sterically like a partial cork in a bottle or so. No, it's just a direct interference of the effect of the binding of erythromycin with the core of the ribosome, the PTC, where the chemical synthesis takes place. And that is what explains this stalling. That was quite a surprise to the field. And we have seen that now in several instances. We're about to write that up, where actually it's not just a simple blockage, it's really an interference with the obviously very sensitive machinery which does the chemistry here, the synthesis. So that is the main result which we got from that study. A short interlude maybe on something which also surprised me quite a bit. I'm not a biologist, I'm a physicist, but I was really amazed to see that there is something which is called frame shifting, and that has been studied again in the lab of Marina Rodnina. And that is, you all know that the genetic code involves triples of base pair, and each triple of base pair encodes one amino acid. So the messenger RNA has to be shifted through the ribosome in steps of 3. Oh, yes, please.
Give more loud.
I think someone had accidentally
All right.
muted themselves. If someone would like to ask a question, please do raise your hand or put it into the chat.
Wonderful, thank you. So the messenger RNA has to be pushed in steps of 3 nucleotides through the ribosome for that the new tRNA can bind with its 3 cognate nucleotides, right? So, but what happens from time to time is that the shift is actually not 3, but maybe 2 or 1 in one or 4, right? And then, of course, the whole thing gets out of register, and you would think everything is lost, right? Then complete chaos. And most of the time, that is actually the case. But bacteria have actually evolved to make use of that. And so what happens is actually, like you can probably see here in the lower part here, should maybe stop that one here. Here in the lower part, that if you shift by 1 at a particular position, which is the signature is a polyala, so 6 or 7 alanines in a row, so A's, sorry, not alanines, A's in a row, that enhances the probability for such a frame shift. And then the code downstream is such that it can produce a protein in the 0 frame register, but also shifted by 1 in the -1 frame. It just generates a different protein. And that is a mechanism to regulate. For example, that is a mechanism to detect the binding of antibiotics. And as soon as you bind the antibiotics, then you get this frame shift. And this frame shift synthesizes an enzyme then downstream, which works against cleaves the antibiotics. So that's a nice reaction in the tug of war between bacteria and natural antibiotics. So amazing in terms of evolution. And of course, now we were interested in how does that frame shift work. And one thing people have done, and also quite excessively in Marina's lab, is to measure the so-called frame shift efficiency for a huge number of different test sequences, like a few of them are shown here. So you systematically mutate all these base pairs. 6 are involved in the actual frame shifts, 2 triples. And then for each of them, you kind of just measure, if you do the experiment many times, what fraction, what percentage actually gets frame shifted and what percentage remains in the canonical arrest. And that is called the frame shift efficiency. And we set out to understand the kind of molecular origin of this efficiency. Now, we had a very simple model in mind, which was kind of an equilibrium model in terms of free energies. So the idea Lars had developed is, okay, if we attribute a certain free energy to the 6 base pairs shifted and the 6 base pairs unshifted, and the free energy difference between the two, then the frameshift efficiency should just be the Boltzmann factor here shown on the left here of this difference. Pretty simple, if everything is in equilibrium. And a further assumption was that this total free energy of the 6 base pairs is just the sum of individual base pair free energies, formation free energies of the 6 individual involved base pairs. In other words, that these energies do not depend on the environment and they don't crosstalk. That's the assumption. And then you just add up 6 base pairs free energies, and that's it. I should probably emphasize here, there is no simulation involved at that point, right? It's just about assuming free energies. And I'm just mentioning that here because what Lars then applied to arrive at these free energies. Now the task obviously is you have these measurements of the frameshift efficiencies and the free energies somehow connect to it, but how and can we derive these free energy differences for the individual base pair from this experimental data? And he developed a Bayes approach, a Bayesian approach with Bayes probability theory or Bayes statistics here sketched on the lower part, where he just wanted to get out the probability given all the experimental data, what are all taken together, all these base pair free energies. And what he got out of this statistics, still no simulation involved, is probability distributions like that. So for each possible different kind of base pairing, you get a certain free energy difference. And I'm showing that to show you that actually these distributions are pretty narrow. So the experimental data suffices to really nail down these individual base pair free energies. And of course, once you have them, you see you have a wonderful model and a wonderful way to predict now frameshift efficiencies. And this is tested here below. So we take a few more measurements, which we haven't used to calibrate our model, and see how well they are predicted. And I guess, long story short, they all lie on the diagonal. X is the experimental measured efficiency, and here is what the model would predict. So we can now predict quite reliably these frameshift efficiencies, and that's also quite nice. There's another story I don't have the time to expand on, which actually addresses the question whether we are really in equilibrium. And the nice story is the whole thing depends on, if you do it very fast, depends on how fast you do it. If you do it slow, then it's independent of how slow you go. But if you go very fast, it starts to become kinetics dependent. And we also include that kinetic term into that model now recently, and that improves the also this timescale dependency quite a bit. So that kind of concludes what I wanted to share with you on the ribosome. And I think I would need like 5 or so more minutes. Would that be okay?
I think that would be great. And there have been several questions. I think it'd be great to hear about the intrinsically disordered protein.
Right. Okay. Yeah. That's actually not a too long story. So you all know that there's a fundamental difference between a folded protein like the one on the left and an intrinsically disordered protein on the right, which simply doesn't adopt one single native structure, the deepest well in the free energy landscape, but continuously jumps pretty fast between all dozens or even hundreds or thousands of intermediate states. So that's what you see on the right side. And actually these IDPs as intrinsically disordered proteins, they are pretty frequent in eukaryotic cells and also have pharmacological relevance. So what we did naively, in order to understand that a little better, we always, and that's something I can really recommend, we always routinely employ different force fields to see if the results depend on the force field. We also did that for the IDP here, pretty simple IDP, and you can see a dramatic difference here between an amber force field on the left and a charm force field on the right. And it's the same IDP, the same sequence, so there shouldn't be really a difference. And that, I think, sent a few ripples of shock through the field, testified by the citations of that paper. And so that motivated us to look a little closer and do quite a large scale comparison. So we teamed up with McCarroll, and who has a wonderful set of proteins for testing charm in place. And we run that for 8 different force fields. One, by the way, I should highlight here, that is by also the absent here by Rohin Papu, whom I like super much. So he does great work on implicit solvent models. But all the others are explicit solvent force fields, and we try to compare those more systematically. And what is shown here is for, again, the same so-called RS peptide, a pretty short, highly charged peptide. The histogram of radii of gyration, just the size more or less of that molecule, again, for the different force fields. And you see they are super compact, like amber gives quite a compact size. And this amber variant, in contrast, gives quite an extended size. And it's a factor of 2 difference between those radius of gyration, meaning a factor of 8 in the volume. And that is really simply unacceptable, right? So you can't have a factor of 2 in the radius of gyration just by changing the force field. Actually, a running gag in our lab was that the force field determines the ensemble much more than the sequence. And that is something we really don't want to see. But that's what happened. And it's not a sampling issue because we were able to provide error bars. So that's a total of 1 millisecond simulation of this thing. So no sampling problems here anymore. It's a force field issue. And that prompted us to improve on the CHARM 36 force field, which you can see here in red. And by comparing to a whole roster of different experiments, so that was a large cooperation between many experimental groups and McCarroll and us. So here you can see, for example, small X-ray scattering on a series of proteins. We did NMR with Christian Griesinger and others, and many other dimensions. And the summary of these findings is shown in this table here. On the x-axis are our 8 force fields, and all the observables we looked at on the y-axis. And the color code is simple. Green is good agreement, red is bad agreement. And you can see one force field stands out. Surprisingly, that's a pretty old CHARM 22 star force field. And CHARM 36, in contrast, which was derived from it, it should be an improvement, actually makes things worse. Performs pretty well for folded proteins, but not for intrinsically unfolded proteins or disordered proteins. So we worked on the 36 and actually improved it to an extent to get pretty good agreement. And that is a new force field, which I would like to advertise here also, CHARM 36M for modified, also now adapted for disordered proteins. And we made sure that actually it also remains its quality for the folded proteins. So we had also a roster of 10 folded proteins, which we checked thoroughly with, again, half a millisecond simulation time. And so that also maintains its good properties. So that's, I would say, together with the amber force field, the most recent amber force field, are the 2 top force fields I would recommend for intrinsically disordered proteins. So that is kind of the story of the intrinsically disordered proteins. And my last story, 2-3 minutes probably, is the dynosome. And I just want to give you a short glimpse because although we started it pretty early, that's a very hot topic and ongoing work here. So the idea behind all that is pretty simple. So in the mid-80s or so, more and more sequences became available, and people started to compare the sequences and cluster them and look for similarities and dissimilarities. And from that, we learned a lot about similar sequences creating similar function and so on. And in the mid-90s, when more and more structures became available, there have been these wonderful structural classifications in families, subfamilies, in folds and superfolds. And we learned, again, a lot more from those. For example, we learned that actually structural motifs and folds are much more conserved during evolution than the underlying sequence. So you can have pretty much the same structure unperturbed over literally billions of years, with the sequence underneath bearing no relation to each other anymore. So structure is the evolutionary conserved motif, in fact. So we thought, why can't we pull up the same trick again with dynamics? So sequence, structure, dynamics, function, that's what we all learn from the textbooks. That's the kind of chain we want to investigate. And obviously dynamics is really the missing link here and between structure and function. So why can't we ask ourselves, are there also dynamical families in terms of dynamics, which may show similar dynamics, despite maybe of totally different structures? Or maybe we have related structures, similar structures, but different dynamics. And how does that relate to function? Does that maybe even help us to predict function? So those were the questions we set out to address on a selected set of something like 100 different proteins. So rather than just doing a simulation for one particular protein of interest, we did a simulation of all those 100 proteins for 100 nanoseconds each, which was at that time quite a bit. Now we repeat the whole thing for 10 microseconds and 1000 proteins, but that's unpublished. But now the question, of course, arises, okay, fine, we have all those simulations. How do we compare dynamics? How do we compare two dynamics, two proteins and their dynamics, which have probably totally different structure? That sounds a bit crazy. And actually it is. And so we pulled off kind of a brute force method and just calculated, don't read everything, I just want to show it's well defined, extracted 34 observables which probe the dynamics. That's all we could find from the literature, and we invented a few more. Just to give an idea, it might be, for example, the eigenvalues of a principal component analysis and how large they grow with the size, the length of the trajectory. It may be the low frequency components of a Fourier spectrum of the dynamics, you name it. Everything we could get a hand on which characterizes somehow a different aspects of the underlying dynamic. We call those 34 the dynamic fingerprint of a molecule. It certainly doesn't capture the full dynamics, but it is a kind of a fingerprint by which you can probably recognize the dynamics. So that's what we have now for every single protein. Now that means that for each protein now, we have the dynamics represented by a vector or a point in a 34-dimensional space, the dynamics space, if you want. And then it's a piece of cake to just calculate, properly normalized, of course, I can't go into that, calculate the Euclidean distance between these points, and they give a measure of the similarity of the dynamics. Right? If it's a small distance, it's a similar dynamics. If it's a large distance, the individual observables are large, and so the dynamics as a whole is also pretty large. So that we used to come up with I jump forward to speak up a little bit, to what we call a kind of a relation tree, a similarity tree in terms of dynamics, right? So that's a high-dimensional data set. Of course, we have to map it somehow down to 2-dimension, and that's one attempt. And each point here now represents one of our proteins, and a connection means they are dynamically similar, and far apart means dissimilar, obviously. There's an example, calmodulin would be over there, arabutocin here over there, and so on. So our question was, how does that relate to function? So there are many different ways to classify protein function. One is the enzymatic database, and they have 10 different enzymatic functions. And if you now map those simply on this tree, you can see nicely that they are not uniformly distributed as you would expect if there is no relation between dynamics and function. No, they occupy certain regions of this tree, like glycosidases, the left half, proteinases in these two branches here, and so on. So now we can do a simple game. You give me a new protein, I do the simulation, I put it onto our tree in terms of similarities to all the other proteins we already have, and then we don't know the function, but I try to predict the function, and I do so by just looking now into my neighborhood on the tree of my new protein and which functional class is here mostly represented. And that would be then my guess what the function of this protein is. And the question is, how well am I doing with this prediction? And that is the result. About half of the proteins I predict correctly. That is much more than you would expect by random, but it would be obviously just 10% or so. And if you do the best structure prediction tool, you get about a third. So when you do dynamics, obviously what is encoded into the dynamics tells us more about structure, about the function of the protein than the structure. And that is nice and actually underscores that dynamics has its right place between structure and function. So it really contains a lot of information about function, and that can be exploited. So that's the main result of that And that brings me also to an end. Here, I just wanted to point out that this field has come a long way since the mid-50s or so, where we started to calculate simple water molecules in terms of quantum chemistry from this simple Schrödinger equation. And so we grew to larger systems, to longer time scales. Recently, we looked at a whole vesicle, a synaptic vesicle in the synapses of our brains. The virus, I already mentioned, it was not the coronavirus at the time, but I'd love to put that up again. It's a little bit too large still. Maybe at some point we will get to bacteria. I don't know where we will end up with, but I'm pretty much sure we are in this field in quite excited times. And I thank you very much for your attention, and just want to put up here the summary slide. I don't need to reread it. That's the take-home messages, and I can probably leave it there while I'm answering questions. Thanks for your attention.
Thank you very much, Helmut. That was fantastic. And you know, I wish we could have the normal applause that we would at this point in time. I would clap, but I think it would sound sad if it was just one person. In any case, it was great. So we have a couple hands up, and I also have a question I hope I can get to. But let's begin with Jan Kirschbaum.
Hi, Jan.
Hi. I have a short question to the IDP part. One is, why exactly are they so difficult to simulate? And the other one is, the force field you found, is it empirically better, or do you know why it is able to simulate the IDP so well?
Yeah, two very good questions I didn't have time to expand on. So first off, I think we can be pretty sure that, you know, the fold of a protein is stabilized by opposing forces or thermodynamic quantities. One is the entropy of the protein chain, which wants to expand the protein, unfold it. And the other is the enthalpic contribution, stabilizing interactions, H-bond salt bridges, hydrophobic patches, you know, you name them, or hydrophobic core, which wants to collapse a protein. Now, on top of all that, and we have studied it and published it also quite recently, comes the thermodynamics of the water shell, which plays a crucial role in this whole stability business. And that is governed by the interactions between the water and the amino acids on the protein surface. And we are pretty much convinced that it is this interaction which is crucial. If you change that a little bit in the force field, then you get immediately larger or smaller. We also know that because, for example, from small X-ray scattering studies, we know that the radius of gyration of many IDPs is very sensitive to the salt concentration, and of course also to temperature and so on. So I would say the reason why it is so sensitive is that actually IDPs are super sensitive things, right? So they react super sensitively to small changes of the interaction between protein and water. So if you put it a bit more euphemistically, IDPs are much better force field test systems than folded proteins. So that would be the positive way of getting it. Now, the second question, what's better in CHARM36M? Actually, we did find a quite artificial tendency of CHARM36 to form right-handed helices. That should not be, right? So helices should be left-handed. But if you let IDPs run with CHARM36, in contrast to many other force fields, you find like 10% wrong helicity in the helix, which form transiently. Whereas in the PDB, you almost don't find them. So that is suspicious. Of course, nobody really knows the fraction of wrong-handed helices, although of course there is CD data which tells you something, but it is quite suspicious. And we found actually at least one bug in CHARM36, which had to do with the radius, with van der Waals radii of carbon beta atoms. So that was something which we fixed. And there were a few more fixes where I think we know what we were doing. I don't have the time to summarize all those, but this C beta, if my thumbs are a peptide bond, and the one hand would be the C alpha and the other would be a connected nitrogen, then we have this dihedral rotation, the phi angle here. And if you now fiddle around with my index finger C beta radius, you can imagine that this rotability due to the van der Waals radius and the interaction between those two is kind of inhibited or changed. And that was one critical thing which was not quite right in Charm 36 and which is now much better in Charm 36M. Does that?
Prabal, oh, sorry.
Yeah,
Yes.
that answers. Thank you very much.
Thanks.
Prabal, you also have a question?
Yeah, Helmut, very nice talk.
Thank you.
So I had a question about this, your first part of the rupture force versus loading rate.
Right.
In simulation. So you seems to be getting always a monotonic dependence.
True.
Meaning the rupture force is increasing as you increase your loading rate.
Correct.
Are there any instances you have encountered where you see a non-monotonic dependence? Because that also reflected in your energy landscape. You have only one kind of a well. That's probably so any other instance, like when we did some simulation with the GP120 CD4 unbinding, so they are actually depending on the loading rate. You see two distinct regime, a slow increasing and a fast increasing. So I just wondering if there, in this case, whether if you go to smaller loading rate, whether you will have any non-monotonicity or you will continue to see this what you have.
Yeah, so in by far the most systems we have looked at, we do have the monotonic trend, faster pulling, higher forces, which also seems to me quite natural. There are, however, what is called catch bonds.
Yes.
And for those, we didn't see it in our hands because we didn't look at this, but other groups did see a non-monotonic behavior, which is the, if you want, the fingerprint of a catch bond. And what happens there is that if you pull fast, then you get a different unbinding path compared to, with a different barrier compared to slow pulling. And that's what happens in a catch bond. So it is possible, but rare.
I see. So I have another question, just for this free energy, what you are calculating from these, I mean, and the Hamar Jabo or the Dotka Jabo. So they are the, in your free energy, I see that the reactant or the product basin or the folding unfolding, if I unfold, that basin is not very clearly visible, correct? So at the transition state, how do you exactly calculate that? That's what. So like in this plot where you have the free energy as a function of distance, the transition is not very clearly visible, correct? The one.
Okay. You mean this plot here?
Yeah.
And you mean by transition this maximum here, or?
Yeah. So meaning if you are pulling at different rate, so here you are probably not showing that. So I mean, if you do it for different rates, the transition. So is that the two basins, binding and unbinding? They are two so well separated, well with a clearly nice barrier. That's what I'm trying to figure out.
Right. So I fully agree. This maximum is not really a maximum. It's just a kind of a hump, right?
Correct. Correct.
But notice, I just tried to see whether I can annotate this here.
Because in such a case, using the transition state, what Javo and Hamar, so that may not be fully compatible. That's what I'm trying to figure out.
Yeah. What I wanted to draw, but I understand I cannot draw here in this setup. If you start pulling, then you apply an additional energy, and that energy looks a little bit like that. It is downhill on this plot here. And the faster you pull, the higher the slope of this downhill energy. That's just a representation of the force by which you're pulling. It implies a potential. Correct. And if you now just try to, in your brain, mentally add a downhill with this curve here, you will see that this develops into a barrier, and also this one. So as soon as you start pulling, this energy develops barriers, and that's also part of the theory which we developed and also which Hammer developed and others. So we do barriers. I should re-emphasize the dashed line here, the theory line, does not come from any of those theories, but just from a pure numerical simulation of Smoluchowski. So that doesn't care whether there's a barrier or not.
I see. No, in the transition state, what I'm trying to say that, as you rightly pointed out, that if I keep on like increasing my loading rate, so that barrier will keep on going high and up, correct? And then if I lower my loading rate, that barrier will come down.
Yeah.
That's what.
And also the transition state will change.
Correct. So that's transition. So, but then
exactly.
unless I clearly define my, so just question is that to settle my question, how do I clearly define the transition if the barrier is not so well defined here? That's what in the present one. Maybe in the different loading rate, you may have a very clearly well defined.
Yeah. Let me re-emphasize that for the theory, we don't really need a transition state.
I see. Okay. I see.
And
okay.
if you have a very slow pulling, then the transition state is right here. Because then you only have a very shallow slope. Whereas if you have a fast pulling, then at some point, the transition state, the highest barrier, becomes this one, because this one is lowered then by the force. And then you get the transition state here. And actually, the distance between the transition state and the well here determines the slope. So that is the reason why for fast pulling, we have a high slope on this logarithmic scale. And for slow pulling, we have this low slope here on that side, right?
Okay. Yeah, fine. I was just trying to see the curvature, how exactly it comes, but it's, yeah, roughly. Okay.
It comes from the, what we call the rupture point. It's not the transition point, it's a different thing, but that also changes. And so does the rupture distance. And it comes from the difference between the minimum here and the point of rupture. And the point of rupture is left of the transition state.
Okay, thanks.
Sure.
Debashish had also indicated that he's had some questions.
Okay, so first of all, very nice talk. My
thanks.
questions are on the ribosomal frame shift.
Okay.
I have two related questions. So in this, equilibrium free energetic calculation, so there the role of the pseudoknot is not included, if I understand correctly, right?
Oh, thanks for the question. Yeah, I didn't talk at all about the about this thing here on the right side here. That actually, it's pretty tricky because that affects the kinetics. It slows down the whole thing. And our theory, this simple version, which I showed here, works only for, as I said, for the slowed down. In that sense, the pseudoknot's effect was taken into account. If you remove this pseudoknot, the frame shift efficiency changed. And in our kinetic model, which I didn't have time to present here, which also involves barrier heights, not only free energy difference between wells, there we can see this effect nicely. And we are just collaborating now with Marina in order to really get the whole range of speeds by modification of this pseudoknot here. Thanks for the question.
So that's what I anticipated. The kinetics is important as far as the role of the pseudoknot is concerned, because depending on the strength of the pseudoknot, it weights and likelihood of frame shift increases. My second related question is, because that is something that simulation can only probably answer, that right after -1 frame shift, it again starts as if at that point it was waiting longer, but once it has shifted by -1, it is easier for it to move forward.
Correct.
So what changes take place there after -1 frame shift locally?
I'm not aware of many changes. It's a very good question, but I think we would have to look again at this pseudoknot in order to understand why it's speeding up. And if I would speculate, it should be about at that position where the pseudoknot gets dissolved. So I would guess it can only move on after the pseudoknot is dissolved, and that is the rate limiting step here. But that's a speculation. I don't know. We would have to simulate that, but I think that might be pretty hard.
Yeah, the reason I'm asking this, that Atkins has given some summary of 5 possibilities and so on. I have seen in his also collected this book. So I was wondering whether out of those 5 possible scenarios, any sort of one of them seems to be most likely or not. What is causing this frame shift, and after the frame shift, what causing it to resume its journey?
Right. So what causes the frame shift? I think that is pretty clear. It's the fact that because we have this poly A, the barrier between separating one register from another is pretty low. So that's what causes the frame shift. If you don't have this poly A, almost no frame shift.
No. So my question is a little more detailed. So say during this elongation cycle, so in which particular stage this happens. See, the cause is known, poly A is responsible, but in which stage of the elongation cycle it happens. So there can be at least 5 different, if not more, possibilities.
Oh, okay. So I don't have a slide with all the stages here, and I'm certainly not a ribosome expert, but I understand it's the stage where the 2 tRNAs are still in A and P, but the cognate sites are already shifted in P and E. So that's the so-called hybrid state. So that is the state where this dynamics presumably goes on. But I should emphasize that's not the result of our simulation. That is what I learn from spectroscopy done on real ribosomes, right? So that's not our results.
So it's the hybrid state in which it happens, not in the classical state. That's what
the idea is. But I'm not sure if it's really proven.
Okay. Okay, because I thought it's still not resolved, and your simulation probably can resolve it. That's why I was asking. Yeah.
We can't, because there is no simulation currently, right? So we would have to set up atomistic simulations in order to address that. And I figure it's not so easy.
Okay. Okay, thank you.
Mm-hmm.
If I can maybe ask a quick question of my own. On the Dynasome work, when you're trying to do the function prediction, so you've based everything on this kind of graph analysis, neighborhood analysis, but I wonder, you have this great feature set you extract from the simulations. Have you tried just training a random forest classifier or something like that to do this, the function prediction instead? Because it could use that feature space probably very efficiently.
Yeah. Thanks for that question. We tried everything we could get a hand on. I should, however, emphasize that at that time, we had like 100 points in a 34-dimensional space. And so it's not really a densely populated thing, right? So you very easily believe you see a cluster where there's actually just a accidental getting close of 2 proteins or something. So it's very easy to overinterpret. And actually, we had a collaboration with mathematicians from Zurich who also saw one of my talks, and he said, give me that data set and I find you the cluster. And I did, but I cheated a bit, I have to admit. I didn't give him the original data set, the real data set, but I did a principal component analysis on our data set. And then I used that. You know that principal component analysis essentially is a model of a multivariate Gaussian, which best fits the data set. Very much like a variance and an average fit one-dimensional data set. And so I used that multivariate Gaussian extracted from the data set to throw random numbers according to this Gaussian distribution. So just by construction, there cannot be a single cluster in there, right? And so I sent this to him. And he found pretty many clusters, as well as in our real data set. That ended the collaboration and our friendship a bit, but it showed us how easy it is to get misled, to find clusters where they are not. That's one of the main reasons why we went now to more and more proteins, and also to better, less brute force collections of observables. Markov models play a central role here.
That's very interesting. Thank you very much.
Sure.
So I think we are at time now. If anyone has any other burning questions, maybe we could address one more, but otherwise we'll just adjourn, I think. Anything else? All right. Well, in that case, thank you very much again. It was a fantastic talk and I think a great discussion. And thank you, everybody, for participating.
Many thanks for joining in, and Debashish, many thanks, and Peter, many thanks also for sharing. Yeah, very grateful for the opportunity to share a few results, and have a great day. We'll meet in Zoom at some point.
Thank you, Helmut. Thank you, Peter.
Thank you.
Great time.
Bye.
Great.